help you understand who, because it defines a lot of times who you are. I mean, at least in Indian crowd, a lot of it. What touched your heart is what I want, what I think everybody should know. Okay, that defines you guys as a person. And what's more important than really getting to know people at a personal level? All the shallow hellos and highs and oh my God, you're so good is, not useful. When you know people personally, you make great friends, right? And you know vulnerabilities of people. I'm sure I've told you a lot of my vulnerabilities, right? Being honest and open, that goes a long way in making personal connections. So I want you guys to do that tomorrow, okay? Make that personal connection with each other. That will help you. You guys should be friends for life, okay? Just because you spent 6 hours, not because you're in the same university. Some of you came from outside. I actually really appreciate you guys who came from outside for this course, okay? Thank you. The rest of you are just forced by your teachers, like, yeah, go do the class. Just kidding. Okay, all right, good. Huh? Good? Thank you. So movies tomorrow morning, okay? I'll tell you all about my Shah Rukh Khan movies. Okay. So histone variants, right? We're talking about histone variants. There are multiple kinds of histone variants. This table lists them. You guys can read the papers again. I don't know to like go by each one by one. They obviously have important role, as I said earlier. Cells trying to use these variants to tell itself, to prepare itself for later, to tell its genome, right? What to do, what to mark, how to prepare yourself for bad weather, rainy weather, right? Or for normal purposes. Okay, this is going to be a recap of everything that we have learned, but in a different way. Okay, there is nucleosome, that is the gene, there's a transcription start site, there's a word called NFR. How many of you can guess what that means? Oh man, it's written there. Oh, so sad. Nucleosome free region. We need nucleosome free region at the transcription start site. I'm sure you've got that. You must be dreaming of it, right? At the end of the day, you would be like, I know nucleosome needs to be moved. Right, good. So, a nucleosome free region is found at the beginning of the transcription start site and at the end of the transcription start site too. Why? Why do you need it? Why do I need it at the end of the transcription start?
Transcription terminators have to come and bind with it.
Wonderful, thank you. I don't know if that is true. I'll accept your answer. Okay? Because somebody has to come there and bind, and there is a co-transcriptional translation that goes on a lot of times in non-eukaryotes, in prokaryotes. In eukaryotes, that doesn't happen that well, but does a lot of post-transcriptional processing things happen where the factors. Your name is Soumya or Adya, or what is your name? Adya. Adya said the other transcription end side factors come and talk and help out. There's a lot of post-transcriptional regulation that happens. For you guys know polyadenylation, I actually don't know all of it because I don't study it. I don't remember them. Okay, but you guys can read about it. So I'm not going to pretend to teach that. Anyways, enhancers have nucleosome free region. That's how they were discovered to begin with, forever. People did these things, and I'll talk about this in a second. There's an enzyme called DNase 1. What does it tell you? DNase 1. By name, DNase 1. Yeah. It cleaves it. It has a specific pattern it creates, especially in the open chromatin. Before ataxia came, it was all DNase 1 sequencing. Things called DNase 1 hypersensitive sites, DHS. That's how all enhancers were discovered. First, initially. Then they figured out, when histone modifications came along, that histone H3K4 monomethylation and histone H3K27 acetylation actually map very well with the DNase 1 hypersensitive sites. So said, boom, they must be marking enhancers. Got it? DNase 1 why DNase 1 is an easy enzyme to get. Our research was not advanced 30, 40 years ago. This is how people started, right? Earlier, a Nature paper used to be cloning a gene, which is like a, daily affair these days, right? In labs, like ours. Like I must have cloned like 1,000+ genes with my own hands. Okay, so that's that. DNA is one, and NFR, the nucleosome free region, has transcription factor, recruits switch NIF complex, recruits histone acetyl transferases, marks the initiation site for acetylation, right? Goes on, acetylation sites recruit Tata binding proteins, RNA polymerase that needs to start the pre-initiation complex that it goes through, the elongation happens, acetyl with the in front of this the RNA pol, there is an acetylation. Behind it, acetylation gets wiped out. Acetylation doesn't remain on the elongation sites. Okay, the rest of it does. There is a H3K36 marking that happens, etc., etc. More importantly, the chaperons come, put the histone variants in the beginning, as I said, H2AZ. They put it afterwards to H2AZ and H3.3, but H2AZ is then removed through InnoAT. I told you guys before we left. Okay, did you guys remember that? Right, InnoAT removes H2AZ and then H3.3 remains. That's a memory. Okay, cool. That was a recap. Histone chaperons. Chaperons, I showed you this picture earlier where we started about telomerases and the lifespans and all the FDA approvals of the drugs. There are different kinds of other kinds of histone variants that are found at different places. Do you guys know the centrosomes? What are centrosomes? Junction here. This is centromere. Sorry, centromere as centrosomes. No, centrosomes are different. I'm sorry, I misspoke. Centromeres. You guys know centromeres. You guys know telomeres. Centromeres are where it's the middle or somewhere in the middle. There is some region in the chromosome where there is not a lot of genes. It's just centromere, which is where a lot of where the centrosomes, so they pull them down from and all of that, right? Cell division. Do you remember cell division? I don't, but you do. Okay, good. Centromeres, around them are heterochromatin region. This is called pericentromeric heterochromatin, another part of chromatin structure, if you will. Okay. There's telomeres. Telomeres have heterochromatin in them, except those repeats and the TERK and the corresponding enzymes. Okay, cool. So the heterochromatin is found in pericentric heterochromatin at the end of the telomere, at the end of the chromosomes and telomeres, that protects them. Telomere protects the decay of some chromosome. Is that true? Probably. I don't know. It does. Okay, good. So they have a different structure. They have different histone variants that go on. ATRX cooperates with DAX. DAX puts on histone H3.3. Okay. ATRX is mutated in over 30% of all sarcomas. What are sarcomas? Sarcomas are cancers of muscle-derived or mesenchymal lineage-derived cancers are called sarcomas. Over 30% of many sarcomas contain ATRX mutation in them. Pediatric gliomas. Gliomas are brain tumors. A lot of brain tumors contain ATRX mutation. Pancreatic neuroendocrine tumor, Steve Jobs died of that, right? Contain ATRX mutations. Okay, cool. ATRX is actually a chromatin remodeler. It talks to DAX. It's an interacting partner, very close interacting partner, husband-wife, DAX. DAX. DAX is the histone chaperon for histone 3.3, works on pericentromeric heterochromatin. Okay, centromere 3. And it multiple types of heterochromatin. CENH3 is actually only present in centromeres. Okay, that's a specific type of histone variants of histone H3. And there is a specific chaperon called HJERP that puts that on. Okay, I know nothing about HJERP, to be honest, so you guys can figure it out. HERA and SR cap and others are more important in the active regions, the euchromatin part of things. Okay, so we'll talk a little bit about a couple of those. Got it? So there are multiple kinds of histone chaperons. They're specific to specific histone variants. Histone chaperon, histone variants, right? And there are other chaperons to put on and off, just regular nucleosomes. Okay, cool. Got that? Was that confusing? Did I speak too fast? One of the criticisms is I speak fast sometimes. I do. That's a criticism I get every single time from every single audience. My my, what is it called? Excitement makes me do that. So I have to tell myself to not speak fast. Okay. The point of the slide is there are histone variants and there are histone chaperons. You don't need to remember who does what. That's not going to change your life right now. Okay. What it is important is there are specific histone chaperons for specific histone variants. Histone chaperons put on histone variants on the nucleosome and remove them. That's it. That's all you care about. Okay. You can remember ATRX if you want to. Okay. This is another picture of similar kind. I just like to pick pictures from review articles of different kinds. Everybody, because all scientists have different way of expressing their knowledge. Like I'm doing it to you, there'll be somebody else express their knowledge in a different way. They write review articles, and that gets you thinking about how people think, because review articles do not just summarize the findings. They also put their perspective on what does that finding mean. How do they view science? What I am telling you, how I view science, may not necessarily be exactly same as another epigeneticist out there. So it's important to have all of you scientists think about science together, because together we solve it. Okay? I might be wrong. You may be correct. I may be right about something. You may be correct about something else. Okay? That's why the review process of papers has multiple scientists in them, right? Peer review is very important because we need to get perspective that is consensus among everybody. No one person is right. That's why I need criticism, right? Because I'm not going to be right all the time. Okay, so this slide lists them. I think you guys got the concept: centromere, pericentromere, replication sites, DNA damage sites. Okay, so actually it's important. It's important that replication sites have different kinds of histone variants on them. They have a different process. Transcription is a different process, right? And DNA damage sites has actually a different kinds of variants that get utilized there, okay? DNA damage is a whole world on itself. There are hundreds of scientists who work on that. It's a very, 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 very important world, right? But I'm not going to tell you that. I'm not going to teach you that because I don't know enough. That's not the subject. But you, if you're interested, read about it. Watch YouTube videos. I'll keep repeating that. Okay? Cool? Okay. So this sort of summarizes where everything about where what is used. I'll come to a couple of these in a second so that you can understand a little bit. Okay. And you don't need to note everything, right? But it is. So like, for example, histone recycling happens after DNA replication. We talked about heritability. I really didn't get a lot into it, but that's something which is important. I'll show you a couple of slides on that later. DNA damage repair, 3.3, 3.2, 3.3. These are different ones that are used at different times. CentP on centromeres, we talked about that. In transcription, we talked about 3.3 and H2A point Z, very important, right? And in heterochromatin, there is 3.3. Okay, cool. Did I show you this? I did. Sort of. Sort of. Okay, so let's just repeat this for a second. There is promoter region, there is coding region. Promoter region has AT rich sequences. You need to look at this carefully, right? The legend, because otherwise you'll not get it. I will not get it. Whew. On a -1 position, there is an H2A. Normally, what are they trying to show us? Oh, they're trying to show us the region that will become nucleodomes depleted region. Risk comes in, binds to nucleodome depleted regions, goes away. Risk, when it binds, puts on H2A point Z. Where is it? They don't show that here. I thought they showed it here. Oh, here it shows that on the bottom. So replacement of 3.3 H3H4 by 3.3H4, meaning it's a different nucleosome. H2A, H2B regular dimer by H2A point Z, H2B dimer, okay? By SWR1 and HERA. These are two different chaperons for a specific function. HERA does 3.3, right? SWR does SWR, that is associated with RISC, the RSC complex actually, okay? RISC comes, removes one of the nucleus and opens the thing for the makes a nucleosome free region. That was the point there. I'm sorry. Then risk removes this. Hira puts on the 3.3. Swar puts on H2A point Z. Now the gene is marked for activation. Okay. And then when the things go on and as the elongation happens, InnoAT removes H2A point Z back to the regular H2A. Okay. Okay, that was all that I think was important there. Let's not go crazy. I think you guys will get confused. In the heterochromatin region, this is the heterochromatic region. There's H3K9 trimethyl. We know heterochromatin has H3K9 trimethyl. Now you would know by now, right? ATRX comes with DAX. It also interacts with SETDB1. Remember SETDB1? SETDB1 had an MBD domain, methyl binding domain, and had a set domain, puts on the H3K9 tree. Cap1 is actually one of the TRIM proteins, TRIM28. That's a transcription factor, very important to setting up heterochromatin. We didn't go into this, but just so you know. DAX replaces 3.3 K9 monomethylated 3.1 with the 3.3 that is methylated at monomethylated H3K9. Okay, it doesn't matter for you. It just marks the region, and then K9 mono will recruit, if I'm right, HP1 dimer. HP1 will recruit SUV39 through H1 and H2. I told you this earlier. That will mark more K9 trimethyl that will spread the chromatin. So that's a seeding mechanism for spreading chromatin. Heterochromatin. Sorry. Did you guys get that? Should I repeat that? I should repeat it? Okay. Let me try to do a better job on this. Okay. So once, let's say, part1 is done transcribing and we want to silent that region for H3K for heterochromatic region. Okay. What DAX, ATRX DAX is going to do is it's going to put on, it will remove the regular 3.1. The regular one is 3.1. Okay. The regular H3. It will remove 3.1, which is blue, with red, which is 3.3. That is specifically monomethylated HK9. It's monomethylated by SETDB1. That's why they're showing you monomethylation. Okay, this monomethylation will allow HP1 to come and bind to that place, recruit SUV39H1 and H2, which is another histone H3K9 trimethyl transferase. Okay, and now that will spread the chromatin. So you saw there was no, this was open region, right? Now you have K9 trimethyl marked nucleosome here, right? Once the reaction happened. Did you get this? So on top, this is open and it has part2 and RNA, but now this has K9 trimethyl that was marked by SUV39H1H2. That was recruited by HP1. Okay, cool. HP1 bound to K9 monomethylation. K9 monomethylation marked 3.3 was put on there by ATRX. ATRX had recruited SETDB1, which allowed that K9 methylation to be marked. That's the process. Okay, that's the process of heterochromatin spreading. Okay, cool. All right, got that. So now we know how histone variants work, how histone chaperons use them in the process of building chromatin structure. In this case, it was building a heterochromatic structure in an active region. So that's a epigenetic process. Okay, cool. We got this? All right. This slide just lists other kinds of histone variants and their association with transcription. Okay, so on the bottom, you see expression association. When H2AZ is there, we know H2AZ is a positive influencer. It's absolutely needed. It's a needed chromatin. Macro H2A, which we didn't talk about, is another kind of H2A histone variant that essentially is associated with positive or negative gene expression. Macro H2A needs to be evicted for expression to occur. Okay, so when macro H2A is there, transcription does not occur. It's usually used for silencing. We actually don't know enough about macro H2A. Okay, it's a larger variant of H2, so it is more bulky. It's bulkier. It's not just one or two amino acids. It actually has a large number of amino acids. There are others such as H2A point B, which help in the splicing. And you guys know about mRNA splicing? Okay. Epigenetics also plays a role there, which I didn't go into. I wish. And 3.3, we talked about it has positive and negative influence both. It is there on the heterochromatin, which we just looked at, like in the previous slide. And 3.3, we also looked at in the gene body, where it creates a memory for transcription. Okay, cool. Good. That's all the knowledge I wanted to give you there. Okay, histone deposition during replication. And to be honest, I didn't know enough about this myself. So I was reading papers this morning to think about what does it do. So I put this text on the right. I was like, I'm not going to remember this. So you will have slide, you can read about it. But basically, when the replication happens, I think we're going with too many things, right? And I'm just trying to cover too many things. And I know that's something I should not be doing, but I will just do it for next 2 slides. So if you guys want to know this more, you can go look up. I don't expect you to remember it, so don't look at me like, okay? Okay? Because I didn't remember it either. I didn't know. I mean, I've listened to talks, I've heard talks, I've listened to, I've read papers, but I can't remember it. My brain is not good enough for that, and I don't expect you to do that because you study way more than I do. You're studying plant biology, microbiology, right? Recombinant DNA technology. So sorry about loading you with lots and lots of information. Don't expect to remember it. I will send you the slides. Okay? You can look at it if you want to. Okay, so in this particular case, during DNA replication, there's a leading strand, there's a lagging strand. Did you guys know that? Okay, I had forgotten about it. When I read this morning, I was like, yeah, that's right. It used to be like that. Okay, and there is the region behind the fork, right? This is a fork. This is a replication fork, right? Replication is leading. So can someone actually tell me what happens in leading and lagging? Yeah, go ahead.
There is continuous synthesis in the leading strand. And
Okay.
there is segmented or discontinuous synthesis in the lagging strand because the replication occurs in 5 to 3 direction and the DNA is unbound. These small sections of DNA are unbound at one instance. So that is how it is done. Multiple primers have to be added in the lagging strand. Single primer does the whole work in leading strand. That is.
Oh, right. What are Okazaki fragments?
DNA segments.
Short DNA fragments. I was reading, I was like, what were Okazaki fragments? I was, thank you. See, I can learn from you too, right? I actually had completely forgotten about Okazaki fragments. Again, because I don't work in the field, there's no point me remembering this. Okay, so there is Okazaki fragment somewhere here, right? What this leading synthesis strand going first or lagging one? I think it was opposed. Simultaneous. Wow, you guys know this stuff. Cool. Somebody wants to explain this figure then to me? All right, one of you come up and explain this because I have limited knowledge. It's all written on the right side, and I will tell you what I know, but I want you guys to tell me what you think. Look at the figure and tell me. You want to come? You're Soumya. You're Soumya. Okay, I thought one was Soumya, one was, what's your name? Adya. Okay, I don't know all of your names, guys. Sorry, but I'll know some of you. Okay, come on.
Know about the chromatids that are attached, but I can explain replication.
Go ahead.
Okay, so basically, in replication, the MCM2 which is written here, this is helicase. And PCN ring, this is clamp. And then there is one RF factor, if I am correct, that is clamp loaders, which loads this. So we have one leading strand and another lagging strand. RNA polymerase, sorry, DNA polymerase polymerizes both the strands.
So polymerization occurs in only one direction, that is 5' to 3'. So towards the 4, which is andar jata hua, that is the leading strand, and away from the 4 is the lagging strand. Now what I have in my understanding, मुझे नहीं पता कितना सही है, but the RNA polymerase binds to this primer attaches there, and then RNA polymerase binds. So this fork is opening as we are going ahead. So it attaches from here, and then there is no space for it to go ahead. So it just reattaches, and then there is lagging strands, Okazaki fragments that are bound in this direction, and then it is further ligated.
Great, thank you. What does the figure tell you now? DNA polymerase, not RNA. Yeah, okay. Go ahead. What do you see?
Remodeling complexes, removing the.
What is MCM2? Okay, let's go through this figure together. What is MCM2? MCM2 is the helicase. Helicase is part of? Good. What does the figure show? It
The
show.
nucleosomes are being removed from the segment, from this part of the DNA by the remodeling complexes. So they're removing it from here to make the segment available for replication.
Histone chaperon. Fact is histone chaperon. MCM2 itself has a itself can lead, at least that's what the right side says. The MCM2 subunit of a helicase can bind H3H4 tetramer, regardless of which H3 variant it contains. Okay, and by deposit it directly onto the nascent DNA or pass it on to histone chaperone ASF1B. ASF1B is a well-known histone chaperone. Okay, go ahead. So look, I mean, you can look and read and think. I guess let's do it as class together again, because somebody else wants to come. Tell me. Okay, I think maybe Swami can sit. Let's have someone else come. Come on, someone else. You want to come? Kapil Dev, right? Come. You're like, it's okay. We'll read the English and we will understand the replication mediated histone 3.3 deposition.
तो मेरा इंग्लिश बोलने का
हिंदी में बोलो, कोई प्रॉब्लम नहीं भाई. Science is not bound by language, right? Bolo, go ahead. I'll tell you a story while he reads it. I studied in Hindi medium school, Saraswati Shishu Mandir, right? I had 12 years of Sanskrit. I don't know Sanskrit anymore. But at the end of the day, I studied in a Hindi medium school, right? So like Kapil's feeling right now, you're feeling like, how are you feeling? डर लग रहा है अंदर से. बात कर नहीं. आज की जनरेशन में डर है ही नहीं.
कोई बात नहीं, बस है कि इसको एक्सप्लेन करने के लिए, बोलने के लिए सब कुछ ऐसा समझ सके, बता सके. जिस लेवल का है
उतने लेवल का बताओ, क्या फर्क पड़ता है. एज अ क्लास वी आर लर्निंग टुगेदर. ओके,
रेप्लिकेशन का प्रोसेस है जिसमें कि डीएनए अनबाइंड हो रहा है और अनबाइंडिंग होने के साथ लीडिंग स्ट्रैंड सिंथेसिस, लैगिंग स्ट्रैंड का सिंथेसिस
वो हो गया, वो हम समझ गए.
और इसमें डिफरेंट एंजाइम काम कर रहे हैं और जो प्रोसेस है इसमें हिस्टोन का किस तरह से इसमें जो रोल है इसके आगे पैकेजिंग के लिए उसको दिखाया है.
बिल्कुल. तो पीसीएनए है
जी.
पीसीएनए क्या करता है, कौन बताएगा? सिस्टर क्लैम्प ना. पीसीएनए में क्या दिखाया हुआ है? कैफ1 को बाइंड कर रहा है वो, ठीक है. कैफ1 क्या कर रहा है? Okay. CAF1 is a chaperone assembly factor complex. ये ASF1B से जो उसने ये न्यूक्लियोसोम ये दे रहा है हिस्टोन डायमर, वो उसको ले रहा है पीसीएनए के साथ वो असेंबल कर रहा है नेक्स्ट न्यूक्लियोसोम, ठीक है. वेयर इज द न्यू, सो दिस इज व्हाई दिस इज इंपॉर्टेंट, राइट. न्यू कहाँ है, ओल्ड कहाँ है. न्यू इज ब्लू, डार्क ब्लू, ओल्ड इज लाइट ब्लू. डार्क ब्लू हाँ यहाँ पे है, लाइट ब्लू एंड डार्क ब्लू का मिक्स है कहीं पे, राइट. सो इस लीडिंग स्ट्रैंड में सिंथेसिस हो चुका है, लैगिंग स्ट्रैंड में भी चल रहा है, राइट. दे आर शोइंग दोज. और कोई क्या बोलना चाहेगा. ओके यू गो, लेट हर कम ऑन. I like it when you guys come up and make my life easier.
यहाँ what I
हाँ.
see here is
हाँ.
lacking strand में जो है multiple plant proteins work कर रहे हैं.
हम्म.
DNA strand, single DNA strand, and as the ये जो ASF 1B है ये एक हिस्टोन वेरिएंट को जो है वो लोड कर रहा है इस न्यूक्लियोसोम पे जो कमिंग जो DNA को सिंथेसाइज हो रहा है.
Okay. तो वो यहाँ भी हो रहा है और यहाँ भी हो रहा है, right? That is happening in both leading strand and lagging strand. ASF1B is a 3.1. 3 point. It's supplying the regular histonase 3.1 nucleosome. हमको replication में 3.3 भी जरूरी नहीं है, right? 3.3 कहाँ पे आता है? 3.3 old है. Sorry. Go back.
सर आई एम लाइक थोड़ा सा कंफ्यूजन है क्योंकि लाइक जो नॉलेज हम लोग को मिला कि हिस्टोन वेरिएंट का जो लोडिंग है उसको आई कांट लाइक परफेक्टली कनेक्ट विद दिस एप्लीकेशन.
That's okay. Let's connect together. What do you guys? I mean, मुझे भी कोई I don't remember this. As I said, again, I don't know either. So you do this, you put this on, it goes here. This is H2A, H2B, green, blue is new 3.1. So ASF1B ने नया वाला डाल दिया है. Where is new H2A? I think the
वो स्टैंड शो नहीं किया होगा जो
okay.
आपने बताया था लाइक ड्यूरिंग द लेक्चर कि जो जैसे कोई जो जीन है लाइक वो ट्रांसक्राइब हो जाता है, ट्रांसक्राइब मतलब ट्रांसक्रिप्शन, दिस इज डीएनए सिंथेसिस, सो दैट्स डिफरेंट.
That's different. Don't get confused between transcription and replication. वो transcription वाला खत्म हो गया वो story. अभी replication story चल रहा है, right? Most of you get it.
मोस्टली मैंने बस ये डिफरेंस को स्पॉट किया.
हम्म.
नॉर्मल जो नॉलेज एप्लीकेशन कैसे होता है, लैगिंग स्ट्रैंड है, इस डायरेक्शन मोड है, 5 प्राइम टू 3 प्राइम होता है, बस उतना ही होता है.
Okay, great. Anyone else wants to read the right side and try and explain? Mr. Akhola, you want to come and explain? डरते क्यों तुम लोग? Okay. इसमें इसने पूरे he hasn't said, this is Steve Hennicoff's paper, he hasn't said all things that matter. ठीक है, in the next slide I'll come back and show there's more chaperons than this at play. This is highlighting the role of two of them, FACT and ASF1B. Those are two important ones. FACT works along with NCM2 to replace the old ones and put the new ones on. Okay, ASF1B takes the 3.3 ones, 3.1 ones and gives it to the in cooperation with CAP1 and PCNA. It will put it on the leading or lagging strand. That works both ways. And in the right, that's what it says, no? Okay, so let's just hold on there. All right. And then there is other histone chaperons. RTT 109 actually is one of the most important chaperons that he didn't even talk about. And then VPS 75, these. So ASF1 he talks about, okay? And these newly synthesized ones. So this figure was slightly better to me. So I thought, okay, let's go to this. We can go back there. So if you look at it, this one, for example, right? This is pre-existing one. the two old green and dark red. Do you see the dark red and green, right? Oops, sorry. So ASF1B puts on one of the new 3.1s. So once newly synthesized H3 is put on. So that's the light green. Do you see light green here in the next nucleosome? This happens after PTM, which is a histone acetylation. Histone acetylation has to occur, and that takes away probably the old nucleosome, old histone chaperone. Sorry, old histone variant. I'm sorry. You see, the old histone variant is acetylated. That is bound by ASF1B and HAT1. HAT1 is the histone acetyl transferase that acts here. Okay, so HAT1 acetylates that. That is gone from this and replaced by a new one. So dark green gets replaced by the light green. That's the step number 1 that happens. Okay, is that cool? So I'm just explaining you this. So from what I understand. And then the second copy of that gets replaced. You see the second copy? So this first is that's through acetylation and ASF1B's complex. One copy of the dimer, his H3H4 dimer gets replaced. Then the other one gets replaced. So this from total dark green, it became light green. Okay? And where is newly synthesized H2, H2B? Oh, at the same time, one of the H2A, H2B got replaced, right? So that's a new one. The new one is the lighter. Do you guys see the difference in these two? Yeah, the dark and the light, right? Dark and the light. So this shows that dark green, dark red became light green, light red. But they were not the same one. The one next to each other were not the ones. So that happens through the ASF1B does the H3H4 and VSP75 and NAP1 do the H2A, H2B. Okay, in this case. Are we good with that? So RTT19, sorry, my mistake. RTT109 does the H2A, H2B and ASF1 does, no, RTT109 also does the H3. No, both of them do H2A. Sorry, H3. Both of them H3. And VPS and NAP do H2B, right? Now it's correct. Okay. So I'm also confused. So they both act together, I guess, in one after another. So first, maybe VPS75 and NAP1 come. They replace the H2A, old to new, H2A, H2B dimer. Then the ASF1, RTTO19 come. They replace the old to new H3H4. And that's how that changes. That's all we need to remember, I guess. Okay. And MCM, as we learned in the previous figure, itself can load the nucleosomes, also new octamers, or it helps ASF1B load it on the new lagging and leading strand. Okay. So this one didn't talk about H2, H2B as much. Or is it, or am I missing it somewhere? Oh, yes, it is saying, it's talking about FACT. Okay. FACT and RTT0109, I think, are similar complexes in two different organisms. So yes, it is talking about that. So FACT or the